Hi everyone, my name is Freek van Ede, and it's a true honor for me to be hosting this year's Young Investigator Award session. And to introduce, also as part of that, our two fantastic awardees, Dr. Emily Finn and André Bastos. Great, yeah, so I'm sure we'll do a lot more clapping as we go along, because of course we're here to celebrate. And so I'm doing this on behalf of the CNS Young Investigator Award Committee, which this year was chaired by Catherine Hartley from New York University, and further included Joel Vos from the University of Chicago, Thor Wager from Dartmouth College, El Astri Mamid from Georgetown University, and myself from the Vrije Universiteit Amsterdam, or Amsterdam. And so before we turn to this year's awardee, I just want to stress how all nominees were extremely impressive, and we should all really feel proud as a community to have so many talented young researchers among us. But of course, we're here also today to celebrate two in particular, and two wonderful researchers who really stood out to us and whose wonderful contributions we will celebrate over the coming hour here. Now, as for the procedure, I'm first going to introduce Emily. We'll hand her over the award on behalf of the society, and finally give her the floor for her talk, and then we'll repeat the same procedure for André. We have also decided not to open the floor for questions after the talks. This leaves more time for the talks themselves, and it also doesn't in this way endanger the celebratory spirit of today's event. But yeah, you never know, right? But you're of course welcome to approach the awardees during the remaining time of this conference if you run into them. Okay. So let's first turn to our first awardee, Dr. Emily Finn. So Emily is assistant professor at the Department of Psychological and Brain Sciences at Dartmouth College. She obtained her Ph.D. from Yale University in 2017, working with Todd Constable and Marvin Chan, among others. She then spent several years as a postdoc in the lab of Peter Bandantini at the NIMH in Bethesda, before starting her own lab in Dartmouth in 2020. Now, a red thread in Emily's stellar research program to date is the development of novel approaches that have opened unique opportunities to understand the relation between brain activity and behavior at the level of the individual, as opposed to at the level of group averages, which are kind of the dominant mode in the field. A key example of this is her pivotal work on functional connectome fingerprints as a unique window into individual differences, ranging from differences in fluid intelligence and sustained attention to differences in how we parse naturalistic visual inputs, social interactions, or even subjective experiences, which is also what you'll be talking about today. Her seminal article describing this approach, published in 2015 in Nature Neuroscience, became an instant classic in the field and has already been cited well over 2,500 times. I think what particularly stood out to the committee was the combination of creativity in developing novel experimental approaches, the experimental rigor in employing these approaches, and the clear vision to maximize the potential that these novel and rigorous approaches offer by applying them to a broad range of questions really at the heart of our field. Emily's visionary abilities are also testified by multiple leading perspective articles that she contributed, including her 2023 Nature review, Functional Neuroimaging as a Catalyst for Integrated Neuroscience. If you haven't read it, go read it. Or her 2021 Trends in Cognitive Science article, pondering whether it's time to put rest to rest. Now, while I don't know if it's time to put rest to rest, I do know that it's time to invite Emily up the stage, hand her the award, and hear so much more about her wonderful contributions that will undoubtedly continue to enrich her field and the reputation of her field for many years to come. So please help me congratulate Emily. Okay, this is the award and the beloved envelope. Here you go.
This, yeah.
The stage is all yours.
Oh, thank you so much. Thank you so much for that incredibly generous introduction. I am so honored to be here. I want to say a huge thank you to the awards committee for this incredible honor. And I, of course, need to take a few minutes to thank everyone that helped me get here. So I did my PhD at Yale, and in particular, these guys really took a chance on me. I almost went straight from college to PhD, but then I had a a little bit of an existential crisis and took a 3-year detour, most of which I spent working for a coffee company in Peru. I eventually came crawling back, tail in hand, and Todd and Marvin really took me back, even though I had never seen a line of code before starting grad school, so thanks to them. I then went on to do my postdoc at the NIMH, where Peter Bandatini and his team gave me just every resource I could ever ask for, both material and intellectual, to do really any kind of work that I wanted to do. And finally, I've been so lucky to land at Dartmouth to start my lab, where I'm supported by an incredible group of other faculty members that I honestly consider friends first and colleagues second. I also want to thank my lab at Dartmouth, who actually did all of the work that I'll show you today. These guys are just so brilliant and so much fun to hang out with, and I'm just continually blown away by the caliber of people that seem willing to move to the middle of nowhere in the icy hinterlands and do science with me and with each other. So thanks to you guys. Finally, I've spent the last few years not only building a lab and a research program, but also a family. So I learned about this award 4 days before my daughter on the right there entered this world back in late December and early January. So for all the trainees in the audience, especially women, I just want to be one more role model to show that it is possible to do this career and have kids before tenure. It's not easy. Thank you. Having a supportive partner helps too. So it's not easy, but it's possible. Okay, so this is a talk about subjective experience. And subjective experience is the idea that our conscious experience of reality is not determined solely by the objective external sensory input we receive. Rather, this experience gets constructed via an interplay between that external input and a complex set of interwoven internal processes. And what this means is that even given the exact same input, the experience or interpretation of that input can differ between different people or even within the same person across time. So perception researchers have capitalized on this for a long time using what are called bistable stimuli. So these are stimuli that have two equally possible percepts. And the cool thing about these stimuli is they allow you to decouple sensory input, which is held constant here, from the subjective percept of that input. So whether this cube is pointing down and to the left or up and to the right. Of course, there are many examples of bistable or even multi-stable information, meaning information with more than two possible interpretations that go beyond simple perceptual stimuli. So here's a famous example from art history, the Mona Lisa. Is she smiling? Is she not smiling? What is she thinking or feeling? Many different observers throughout history have looked at this painting and come to different conclusions. And this may be more of a cognitive rather than a perceptual process, like the Necker cube that I showed you on the last slide. But I think on some level, the basic principle is the same. And I just find this phenomenon so interesting. So that's what I'm going to talk to you about today. So here's a brief roadmap for the talk. I'll tell you why I think we should care about subjective experience. Then I'll tell you why I think we should care about the brain for subjective experience. And finally, I'll spend some time talking about new approaches that my lab's been taking to try to understand the neural basis of subjective experience. Okay, so why should we care? I would argue that the fact that different people can look at the same sensory information and come to, in some cases, wildly different conclusions or interpretations of that information is a pretty important issue in our society today. You don't have to look very far to find any number of examples of this. It feels like polarization is at an all-time high. So my hope is that by understanding something about why people are having, in some cases, drastically different reactions to the same information can help us design interventions to reduce polarization. So it's not just societal level issues, so these interpretive lenses, as we might think of them, also have strong consequences for individual people. We've known for a long time that depression and other mood disorders are associated with a negative interpretation bias, so people with or at risk for these disorders can look at information that other people find neutral or even positive and interpret that information in a negative light. And this is just a figure from a relatively recent meta-analysis showing that this general negative bias holds across a wide range of different stimuli, tasks, and ways of measuring it. And this is one of the most striking findings to come out in this area in recent years, in my opinion. So this paper showed that it turns out that the subjective experience of feeling like you were maltreated in childhood, even in the absence of any objective measures of maltreatment, predicts who is likely to go on to develop some kind of psychopathology. And conversely, objective evidence that someone was maltreated, like from court records and such, if that person doesn't report feeling like they were maltreated, that person is no more likely to go on to develop psychopathology than a control subject who has neither objective nor subjective experience of maltreatment. So in my mind, that's a very clear indicator of the power of subjective experience and how important it is for us to consider and study these subjective experiences. Okay, so hopefully if you didn't before, that's convinced you that we should care very deeply about subjective experience. And now I'm just going to spend a couple of minutes talking about why we should care about the brain for subjective experience. And that may feel like a funny point to spend time on for this audience in particular, but nonetheless I think it's important to justify because after all, if we're interested in people's subjective experiences, we could just ask them, right? So what can the brain really add here? So first of all, people can often tell you what they think or how they feel, but not necessarily why. There's a rich literature going back many decades in both cognitive and social psychology showing us that people are famously bad at introspection. So the idea here is that maybe we can use brain activity as people are actually experiencing the stimulus as a more latent readout of which particular moments or features of the stimulus they might be reacting to that are driving them toward one interpretation or another. Second reason to care about the brain is that the brain is causal for subjective experience. So in other words, if we change something about brain activity, we can change subjective experience. And this has been shown using both invasive methods and non-invasive ones. So for example, if you stimulate the amygdala intracranially, you can change valence judgments for affective images. And you can do the same thing non-invasively with neurofeedback techniques, for example. And there's a lot of work showing that you can change, for example, preferences for different faces, even some hint that you might be able to alter people's high-level interpretation of a social narrative using these neurofeedback techniques. Third reason to care about the brain, in my view, is that sometimes the brain knows better than behavior. So in other words, sometimes brain activity can be a better predictor of the impact that a stimulus will have than explicit self-report, especially at the population level. So for example, some of this work has shown that in public health campaigns, you can predict how many hotline calls certain anti-smoking images will generate based on how much brain activity those images evoke, even above and beyond people's explicit ratings for how effective they think the image will be. And similar things have been shown in the entertainment industry for ratings of TV shows and advertisements. Okay, so hopefully so far I've convinced you that subjective experience matters and that the brain has a lot to tell us about subjective experience. And I'm excited to get into a couple of specific studies that illustrate how my lab has been tackling this problem. But before I get into that, I want to zoom out a little bit and make a broader point about the task states that we're using to get these neural measurements. So full disclosure, I consider myself a reformed resting state researcher. A lot of my earlier work was using functional connectivity, often measured at rest, to predict individual differences in behavior, and that was a powerful approach. But there's probably many of you in the audience who never jumped on the resting state bandwagon, and maybe you are groaning. But bear with me, at first glance you might think that rest would be a great candidate for studying differences in subjective experience because people can sort of lie there and think or feel whatever they want. So it gives you a lot of room for variability in subjective experience. The problem, as many of you are no doubt silently screaming at me, is that you as the experimenter have essentially no knowledge of or control over what the subject is doing or thinking about. So we've started thinking about this as a sort of 2D landscape with these properties on the x and the y axes, and then we can sort of think about where existing acquisition states fit into this landscape. So resting state might sit here in the upper left, where again you have a lot of room for variability in subjective experience, but essentially no knowledge of or control over what the subject is doing or thinking about. Then in the opposite quadrant, you have traditional tasks down here in the bottom right, which have taught us a lot about sort of generally how brain function works across people, but they do leave generally less room for variability in subjective experience. Of course, you have a lot more control over what's happening. And it turns out, as colleagues and I and others realized, and many people saying I told you so, that measuring functional connectivity during tasks is actually more unique, more stable, more reliable, more heritable, and more meaningful in the sense that it better predicts behavior than connectivity measured at rest. So again, if anyone wants to tell me I told you so, I will take that. For reasons of time, I'm not going to walk through the evidence for each of these points individually, but if you're interested, as Frank pointed out, I attempted to repent for my sins in a relatively recent TICS article that lays out a lot of this evidence. Okay, and actually most of this evidence comes from these relatively traditional tasks that are not necessarily explicitly designed to pull out differences in subjective experience. So I would wager that the tasks that are gonna give us the best bang for our buck, so to speak, the tasks that are gonna be the best for studying differences in subjective experience are gonna be the ones that start to push towards the upper right part of this space. So one small but powerful tweak that you can do is to have people rest, but just give them these intermittent thought probes every minute or two, asking them to briefly describe what they're thinking about. And this is ongoing work with Hongji Kim and Wani Wu at SKKU in Korea. So it turns out that if you do this, you can improve both fingerprinting, which is our ability to discriminate individuals from one another, and preliminarily our ability to predict behavior. So here we're showing the correlation of a subject to themselves in terms of whole brain functional connectivity profiles in a different run. And on the left of each plot is the correlation of a subject to themselves, and on the right is the correlation between that subject and the other subject in the data set that looks most like them. And what you can see is that there's much more separation between these two distributions in the pair of runs that contain these thought sampling probes compared to the pair of runs that are just pure rest. And what this means, again, is that subjects are more identifiable or more discriminable from one another when they're doing this intermittent introspection task. And here are some results from behavior prediction. So here we're trying to predict two different traits, imagination, mindfulness, and positive affect. And here the y-axis is prediction accuracy in a held-out sample. These are models trained on functional connectivity data. The distributions in peach are when we're feeding the model data from these thought sampling runs, and the ones in green are using data from rest. So again, same exact individuals, same target behaviors that we're trying to predict, but we can see that this small tweak of just adding these thought sampling probes gives us more accurate predictions. Okay, so we might place these types of thought sampling tasks here to the right of pure rest because they still allow for basically just as much variability in subjective experience, but the difference is that you're drawing people's attention to this experience and getting readouts of it, and that gives you at least some knowledge of what's going on. So we could start to push even further into the upper right quadrant using these so-called naturalistic tasks, like watching movies or listening to stories. And these are really powerful paradigms because everyone is exposed to the exact same sensory information, but they might have different experiences of that information. A couple years ago now, in a head-to-head comparison, we found that functional connectivity measured during movie watching outperformed connectivity measured during rest for predicting both cognitive and affective behaviors. So here again, I'm showing prediction accuracy on the y-axis, and this comes from predictions of a sort of composite cognitive ability score. And we're pitting models trained on resting data in dark gray against models trained on movie watching data in purple. And you can see that pretty much all movies do better than rest, although not all movies are created equal. And it turns out that the content of the movie really matters. So the key thing here is that each of these 4 movie watching runs was comprised of several different individual 3 to 4 minute video clips. So when we broke things down by individual clip, we found that the clips with more social content, which we proxied with this simple measure of just the % of time points containing at least one face on screen, those were the clips that did better for predicting behavior. And we thought that this might be because social content evokes some of the most variable interpretations, so it might help amplify those differences in brain function that our prediction models are capitalizing on. So my lab has gone pretty heavy on these tasks in the last few years, inspired by a lot of foundational work that came before us. This slide is just a brief sampler plate of the ways that we've been using movies and stories to get at some of these questions. So a few years ago now, colleagues and I showed that subjects with higher trait paranoia have more stereotyped activity patterns in certain brain regions while they're listening to a deliberately ambiguous social narrative that we designed to produce suspicion in some subjects but not others. More recently, my fantastic grad student, Clara Sava-Siegel, used fMRI data from 4 short films to show that subjects with more similar neural event boundaries as they're watching the films also arrive at more similar interpretations of the films. And finally, another fabulous grad student, Tommy Botch, used data from 4 audio podcasts to show that neural representations of the concrete abstract axis are unique to individuals, and that somewhat paradoxically, it's actually the representations of concrete rather than abstract concepts that are most individuating. Okay, so I'm obviously a huge fan of these paradigms, but one challenge with movies and stories is that because they're so complex, it can be hard to pinpoint exactly which of their features people are computing on, so to speak, to give rise to different subjective experiences. So in the remaining time, I want to zoom in on an example of how we're complementing these efforts with very carefully crafted stimuli, where we can vary something parametrically about the stimulus itself and determine how that affects subjective experience. And the question we're going after in this line of work is this very basic, but I think also fundamental question of what makes something social. And this may seem like one of those things that we just know it when we see it, but it turns out it's not so obvious. So let's take, for example, these classic Heider-Simmel animations. So even these simple geometric shapes, when you just look at these in a static context, they don't appear agent or human-like. But once they start moving in certain ways, most neurotypical people will very automatically and spontaneously attribute goals, beliefs, desires, social relationships. They'll sort of construct a whole narrative around the movement of these shapes. And we can contrast animations like the one on the left with ones like the one on the right, where the shapes are moving sort of seemingly randomly or just mechanically without agency. And this phenomenon is so robust that these animation tasks have been used for decades. They're often used as social localizers in neuroimaging studies. But in our mind, the problem here is that these stimuli are almost always handcrafted by human experimenters to look either obviously social or obviously random. There's no objective definition for what makes something social or not. And still, we as experimenters tend to frame behavior on these tasks in terms of accuracy. So if you're watching an animation like the one on the left that I designed to look social and you say it's non-social, you're wrong. Conversely, if you're watching an animation that's supposed to look non-social like the one on the right and you happen to get a social percept out of that, you're also wrong. But who's to say you're actually wrong? There's no real ground truth here. These are just triangles moving around a screen. So our lab thought that maybe it would be more correct to study what people actually perceive as opposed to what we intended for them to perceive. So as a first pass at this question, we turned to data from the Human Connectome project, which contained a social cognition task using these animations. And the first part of this work was all led by a fantastic former postdoc in the lab, Dr. Reka Verrier. So in this data set, there were 10 total animations. The top 5 here were intended to look social. The bottom 5 were intended to look non-social. And I'm showing you the behavioral data here. So you can see that for the most part, subjects got the percept that was intended by the experimenters. But there was also some variability. And in particular, one animation, which was called random mechanical by the experimenters, had the most variability in reported percepts. So using data from that single animation, we grouped people by whether they reported perceiving a social interaction or not, and contrasted activation magnitude using a whole brain parcellation. And we saw that almost the entire brain is more active to information that's ultimately perceived as social. And this is really striking because the stimulus here is exactly the same. So the visual input is perfectly controlled. And the only difference here is whether people have that percept of social or not. So we can directly pit the objective versus subjective labels against one another, and that's what I'm showing you here. So in the map on the right, areas in green are ones where the objective labels, or what the experimenters intended, explain more variance in brain activity, while areas in pink are ones where using the subject's own reported percept explains more variance in brain activity. And what we find is that across much of the brain, including both sensory and higher order regions, people's own percepts do a much better job of explaining brain activity than the intended stimulus label. And maybe that's not too surprising, but I do want to emphasize here that this is not how these and similar tasks are typically analyzed. So traditionally, any trial that didn't conform to the intended label would be thrown out as an error, but that would be discarding the very variance that we're able to pick up on here. So we really think that adopting this perspective that socialness, or whatever else you're trying to study, is in the eye of the beholder, has the potential to reveal a lot of new insights even into some of these older paradigms and data sets. Okay, so we thought this was a good start, but the HCP stimuli were not actually all that ambiguous. They weren't intended to be. They also weren't particularly well controlled in their visual properties. So more recently, we've been extending this line of work to try to develop a set of stimuli that are fully parameterizable to more rigorously investigate social perception tendencies across individuals. And this work is led by an outstanding team in the lab. And so here I want to briefly bring back our paradigm landscape and point out that we're really trying to push into the upper right part of this space. So by going back to somewhat simpler stimuli, we're sacrificing a little bit of that richness of full-on movies and stories, but in a way that gives us these parametric knobs that we can tweak, so to speak, to have more control and understanding over what features people might be reacting to. Okay, so how do we parameterize socialness? I'm gonna give you a brief demo here. So I'd like you to watch this very simple video and try to decide how much you think the shapes are interacting. So specifically, how much you think one circle is chasing another versus just moving independently. Okay, so who saw a chase? Who did not see a chase? Okay. About evenly split, that's good. There's no right or wrong answer, you're all correct. So the parameter that we're controlling here is something called chase directness, which governs how much the predator, so to speak, or the shape doing the chasing is allowed to deviate from a perfect heat-seeking trajectory towards the other shape. And I want to point out that this comes directly from really beautiful work from Brian Scholl's lab. The video that I just showed you is at a sort of intermediate directness of 0.5, where people, again, are about evenly split as to whether they see a chase or not. We can contrast that with a very direct chase, like the one you're now seeing on the left, where most people will agree that black is chasing gray. And also very indirect chases like this one on the right, where many fewer people, although not necessarily zero people, will report seeing a chase. There's a lot less contingency in the motion between those two shapes. And we can create sort of an arbitrary number of intermediate levels here as well. Okay, so as Brian's lab showed and as we replicated, this parameter produces very robust behavioral effects at the group level, where as you increase chase directness, you also get more reports of social interactions. But here we were more interested in the individual differences in percepts. So we can actually look at individual differences in these so-called social tuning curves, which we can parameterize using sigmoid functions. And you can see that while everyone shows the same general trend, people also have different biases and sensitivity levels. We brought people back for a second session a few months later and found that these curves are pretty reliable. So here you can just visually see the same subjects in two different sessions. And the interclass correlation coefficients of the sigmoid curve fits themselves are moderately reliable, on par with many other psychological tasks. So we're hopeful that this task captures something relatively stable about individuals' social perceptual tendencies. Okay, so what's going on in the brain? So here's some preliminary fMRI data from a study using these stimuli. So here we're broadly replicating our result from the HCP data set. So if we model brain activity as a function of that chase directness parameter, sort of the objective amount of socialness that we, as the experimenters, have baked into the stimuli, we don't see a ton of stuff popping up. But when we model brain activity as a function of people's own ratings on each trial, we do see several regions tracking levels of perceived socialness. And the direct contrast of these two shows that ratings are the better predictor. And I think the interesting thing about this map is we're seeing perceived socialness emerge as early as this sort of V5 or MT-like area, even though the visual properties of these stimuli are quite uniform, and we also control for a number of low-level visual properties in the models. And we think that this is adding to a growing body of evidence for what's been called a third visual pathway that's specialized for social perception that starts in V1 and sort of progresses or sends information along the temporal lobe. So again, I just find it really interesting that we're seeing something like socialness, which is typically thought of as a pretty high-level property, emerging this early in the cortical hierarchy. So broadening out a bit, we have this idea that there might be social perception landscapes that govern if and how individuals perceive social information. And this is just a schematic to show you how we're thinking about this. The idea here is that the horizontal axis goes from non-social to social. You can kind of think of the units along this axis being some kind of parameter that governs just if something looks social in the first place, like our directness parameter. But then given that people start to perceive something as social, there's also room for variability in the nature of the interaction they perceive. So for example, on a valence scale from positive to negative. By the way, this is definitely a space with many more than 2 dimensions, but I'm just showing 2 here to help conceptualize what this might look like. So a typical or a healthy social perception landscape might look something like this, where you require moderate doses of evidence to start to perceive something as social, and you're relatively balanced in your percepts of positive versus negative social information. But then you could get various distortions to this landscape. So for example, an autism-like phenotype might be associated with, or might require a lot more evidence to start to perceive something as social. You could also have a negativity bias, which I mentioned at the beginning of the talk, where more of this landscape gets classified as negative. And you could also have combinations of these distortions. So you might have people that are both more sensitive to social information and more likely to perceive that information as negative, which could manifest as paranoia or other psychosis-like experiences. So we've already taken a few steps down the road of testing some other parameters that will allow us to characterize the other axes of this space. And we're really excited to continue exploring this idea of how we can use these individualized landscapes to understand other types of social behaviors, especially those that are relevant for mental health. Okay, so to close, I hope that I've convinced you that we should care very deeply about subjective experience and that the brain has a lot to tell us about subjective experience. And I just hope that we can continue to get creative with new paradigms, analyses, modeling approaches, et cetera, to tease out the neural basis of subjective experience, and eventually that we can use what we're finding to help improve real-world outcomes for both individual people and for society. So I'd like to thank my lab one more time and my funding sources and the awards committee, and thank you all for being here, and I'm excited for Andre's talk.